Hey everyone, welcome to this week's live broadcast of This Week in Science. We are streaming earlier than we normally do. Those of you who are joining us live, we are so glad that you are here to join us. We are with a Blair. It's very exciting. Yay! Everybody tap, tap, mic, all good?
Check 1, 2.
Wee. Yes, looking good there, Justin. Tap the mic. That's right. Let's see. What do I have to say? This is a part of the show that's not really the show, but it's the live part of the show. It'll get cut out because we cut out a lot of things for the podcast to make sure that the podcast is a wonderful, smooth listening experience. If you like the live, make sure that you are subscribed to our YouTube, Twitch, and Facebook channels. Hit those likes, the shares, etc., to make sure that we are out in the algorithms right now. And otherwise, are we good to go, everyone?
I think we are. Yeah.
All right, let us begin this show. All right, we are starting this show in 3, 2. This is TWIS. This Week in Science, episode number 995, recorded on Wednesday, November 27th, 2024. A TWISgiving stuffed with science. Hey everyone, I'm Dr. Kiki, and today we are going to fill your heads with Pokemon, peaches, and psychosis. But first...
Disclaimer. We live in an age where science and technology have changed almost everything. The way we eat, work, travel, communicate, how we entertain ourselves, heal ourselves, and learn about the world. Everything is different than it once was. And if you think it's over, buddy, it ain't even just begun. The coming revolution in AI, the unavoidable impacts of climate change, the new geopolitical dynamics of space will all affect what tomorrow looks like. The way we eat, work, travel, communicate, how we entertain ourselves, how we heal ourselves, and how we learn about the world. But one thing will remain the same. There will always be more This Week in Science coming up next.
I've got the kind of mind that can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Good science to you, Kiki and Blair.
And a good science to you too, Justin, Blair, and everyone out there. Welcome to another episode of This Week in Science. We are here on the internets to talk about science on this TWISgiving of a week. We're giving science. What? Welcome. Let's have a fun show full of gratitude, thanks, and maybe we'll, I brought a story about turkey. Yep, we're going to talk about turkeys. I'm also going to talk about peaches, cosmic ray detection, sterile neutrinos, well, maybe not, and sexual rejection, because that'll be fun. What do you have, Justin?
I have life that was discovered on an asteroid sample, embedded cells telling on you, Neanderthal manufacturing, and does cannabis cause psychosis? Definitive answer.
It's a great question. Would love an answer. Blair, you're back with us.
I am.
Yay. I am so grateful. Yay. I'm glad we were able to make this show happen today so you could join us for the show this week.
Yeah.
What is in the animal corner?
Well, I brought wolves with a sweet tooth. I brought some multivitamins that are worth a darn. And I also brought that story about Pokemon that you mentioned.
Pokemon. Okay, let's go. Let's do this. We ready for a show, everybody?
Absolutely.
Yeah. All right. Before we jump in, I want to remind all the peoples out there that if you would like to get TWIS when it comes out, make sure that you are subscribed to the channels where we are located: YouTube, Facebook, Twitch. Make sure you got your notifications set, those little bells and whistles and all that kind of stuff, because then you'll be able to know when we go live on Wednesdays, normally 8 p.m. Pacific time. But today we've got a little different of our time, but you'll know when we're live because those channels will notify you, let you know that we are here. But if that doesn't work for you, you can always find us by looking for This Week in Science or TWIS Science on social networks and also podcast platforms. And our website is TWIS.org. Please subscribe, share, and let's put all this fun science out there. Woohoo! Let's go do that, people. All right. My first story for the show. All right. This story is, we're going to start with a collaboration, a global collaboration that is collecting data from a detector array in Namibia. It's the HESS collaboration, otherwise known as the High Energy Spectroscopic System or Stereoscopic System, excuse me. And this is an observatory, one of the very, very few, and one of the leading observatories studying high energy, not just high energy, very high energy, VHE gamma rays. So gamma rays are very high energy electrons and positrons, and they are usually released high energy from big things in the sky out there away from us, right? They are released from black holes, quasars. Maybe they are particles that have been released and interacted with other stuff and been sped up and shot places. But these are just bits of energy, particles that are going, but even faster than that. So they're cosmic rays are the really, really high energy ones. Gamma rays are a bit less high energy. So we're looking at measurements of giga electron volts as opposed to tera electron volts when we're talking about cosmic rays or gamma rays. Now, the reason that this HESS observatory is looking at gamma rays is that when cosmic rays that are shot from big energetic things in space interact with magnetic fields, they lose energy. And they can also interact with other things when that happens, and they can have little showering cascades that lead to gamma rays. Or, hi, I'm a cosmic ray, I bumped into something, and now I lost energy, and whoo, now I'm a gamma ray. So these particles decay into gamma rays within our atmosphere, and usually at the very, very high altitude levels of our atmosphere. So the HESS Observatory, in this particular study, isn't talking about gamma rays, it's talking about cosmic rays by inferring their presence based on gamma rays. So the HESS Observatory, it's a bunch of radio astronomy telescopes that are located out in the desert, out in Namibia, and in the Comus Highlands of Namibia, 835 meters below the southern sky. All of these 12 observatory detectors work together to collect any data that they can on these energetic particles that are bombarding the Earth and then decaying into the high energy gamma rays that they are able to measure. By compiling all of the data, the HESS Observatory team, which is an international team and has been working together for several years now to detect all of these showers, the scientists have gotten new information from a giant data set with, thank goodness, new algorithms, selection algorithms. People got smart, computers got smarter, and they're able to obtain data by inferring, like I said, about these cosmic rays in the highest energy ranges, up to 40 tera electron volts, based on their measurements of things that are at lower energy levels. So they measured the energy spectrum of electrons and positrons, and the data that they measured is under a tera electron volt, up to a tera electron volt, which is about 1,000 billion gamma electron volts. It's big. There's a lot. So that based on the spread of the data and the statistical probabilities of where and what particle energy levels they detected, they were able to estimate the ranges of the particle spectrum that's out there decaying. So they took into account noise and other aspects of what's happening up there in the atmosphere, where things are coming from. And basically they're able to say, okay, this is the bottom line for this very long story that has not gone very well in my explaining of it. They're finally able to say, what are the things that are sending these cosmic rays to us? And they were able to say, we can't pinpoint where they are coming from, but based on the energy and the spread of the energy that we are detecting with this array in Namibia, there are a select few high energy cosmic ray creating sources within about 1,000 light years of our solar system. So not very far away from our galaxy when it comes down to it. So our galactic neighborhood very likely has not a ton, not a huge quantity of really high energy emitters, but there are a few out there. And so the next step is going to be determining where they're located. What other things can we use to determine where these high energy particles are coming from in space?
So
I think they need to move to the moon. Like, stop inferring. Yeah, maybe get it directional.
Yeah, so that's part of it, though, is cosmic rays, because cosmic rays are super high energy. They don't interact with a whole bunch of stuff. And it's magnetic fields and light that are responsible for the decay of cosmic rays, of changing their energy and their path. And so it's only when the cosmic rays get close to our magnetic field within its influence and start to decay to lower energy levels, gamma rays, etc., that then lead to further decay, because then they're no longer like so fast and crazy that they're not interacting with stuff anymore. So it's because of our magnetic field, and these detectors need to be placed on the planet. We cannot have space detectors at this point in time to detect these cosmic rays.
And so, like, so what kind of things make cosmic rays? Like, why is this? What is this telling us? What are the options of cool things that we could have discovered that are creating these cosmic rays?
Pulsars, quasars, maybe supernovas, maybe black holes, themselves. We don't, that's the thing. We have guesses, but it's what we hypothesize is that very, it's going to only be very high energy bodies, like impacts that would then result in the release of cosmic rays. It would have to be very large, powerful sources to create the energy levels that are seen in cosmic rays.
And so what's the likelihood that these are being created by something we already know exactly where it is and we know it exists? Or is this the likelihood that this is something that's new to us?
So we have, I believe, with the gravitational wave detectors, begun to triangulate the merging of neutron stars, black holes, of various sizes. And it's because we're able to triangulate the location and pinpoint the location of the impacts of black holes that we, in a couple of instances, have also been able to, at the same time, because it was pointed in the direction of our planet, we were able to actually detect cosmic rays that were emitted by an event at the same time. So it has to be like everything is in the exact right orientation to point cosmic rays and gravitational waves and all the things at us so that we can triangulate. We have to be looking at it at the right moment in time to be able to put all the pieces together.
Interesting.
Yeah, I mean, and cosmic rays and other influence, like there's big questions as to, if things are directed, like a laser at the Earth, what kind of impact would that have on the planet? So there are interesting questions as to, what if we got, a whole bunch of high energy particles bombarding our planet?
Well, the appliances would revolt, obviously.
Obviously.
We all become sentient and start taking their revenge.
Of course.
Yeah. It's also interesting to know how things are distributed around not just our solar system, our galaxy, but our universe. Where are the big high energy things, and how big are they? What is it that it takes to create these very high energy particles? And so, like right now, we're basically we have hypotheses, right, that we're testing, but there's still so much we don't understand about the formation of the universe, the expansion of the universe. The evolution of galaxies. We're still piecing things together, really. And cosmic rays can help tell that story. Yeah. Justin, do you want to talk about something spacey?
Yeah. So, I mean, I guess not everybody heard, but we went up with the Jackson did went up to the Hayabusa 2 mission, went and sampled Ryugu asteroid, brought the sample back to Earth, and we found life on it. Not everybody's talking about it.
How contamination or
yeah, is it real? Is it a real thing? A real suggestion that there was life?
Transported to Earth in a hermetically sealed chamber. The sample was opened in a nitrogen class 10,000 room to prevent contamination. Individual particles were then picked with sterilized tools and stored under nitrogen airtight containers before any analysis took place.
Right.
The samples underwent nano
Claire, you're just laughing there. Oh my gosh.
Demography.
You're taking
and was then
so much joy.
Embedded into a resin block for scanning with electron microscopy. Eliminating any potential for contamination.
Right.
And they found lots of rods and filaments that looked like they were, somewhere around 5 days old.
Yeah.
And the variation in size and morphology of these structures resembled very well-known terrestrial microbes.
Ding, ding,
From
ding.
Earth. Yeah.
Terrestrial life has been found in space.
Yeah.
Because we put it
sample
there.
That was immediately contaminated because nothing on this planet
no matter how hard you try. Yeah.
We are awash. It is actually, I mean, gosh, you would think we'll be able, but here's the thing that I'm thinking too. Like, we already know NASA cannot keep a clean room clean.
Mm-hmm.
This has been a problem. They had this idea. We're going to build our our spacecraft in clean rooms so that when we throw something up, a probe that's going to the moon or the Mars or wherever, it's not going to bring any microbes with it. But they have never been able to keep these rooms completely clean. They found one organism that was converting the disinfectant cleaner that they were using into nutrients. They found another that was eating like
delicious pine salt.
Like life is like, we've talked about this before. Like there's like only one life form on Earth. that we all originate from. And you would think, oh, if life can form on Earth, why doesn't it happen all the time? Why aren't there parallel life branches that are completely unrelated to all the rest, and maybe even have a different form of DNA or something like this? And we're like, yeah, it's all related. It's all the same basis. It's all going to have the same origin. And the reason we've talked about it too, we've discussed this already. There's no niches. There's no niches. There's nowhere for anything to get started that it doesn't become food for something else immediately. Like everything is food all the way down. So there's no room.
So I think this story has been framed wrong. Instead of we found life on an asteroid, oops, it was ours. No. We just initiated panspermia.
Oh yeah.
Again.
No, we have.
Like that's
Mars
what that means.
and the moon.
You went to an asteroid, you picked up some stuff. Oopsie, it has life on it from Earth. That means, oopsie, we left some on the asteroid as well. So
we don't know. We don't know if the sample itself went up with this, or if this did somehow get contaminated with their ultra aseptic technique.
You said it was 5 days old, though, right? So,
well, the sample that they saw was 5 days old, but that doesn't mean anything. That could be, every 5 days you could be finding.
Oh, sure,
it, right?
That's fair. Yeah.
So I'm with you. I think there's a possibility that the sample collector already had microbes. Like 100%, that's a possibility. It may even be the more likely scenario than it came to Earth and got infected, or contaminated. But I guarantee you, when we're talking panspermia, we have to acknowledge just what you're saying. We've already done it.
Mm-hmm.
It's not a hypothesis. We've done it. We've put tardigrades on the moon.
Yeah.
We've got bacteria on every probe that crash landed into Mars.
But let's put this back into the concept of when we put tardigrades on them, we put bacteria someplace. We know that there is, algae or whatever on the outside of the International Space Station, whatever. There's stuff out there. But is it in a dormant state? Is it alive, or is it just preserved?
So
I think those are three very different things. Yeah.
So here's totally right. Here's one extra thing we did learn from this study. That asteroid material is a growing medium for bacteria. Whether it went up or came down and ended up on there, it became home. Right? Like there's nothing soil dependent that on Mars or the moon that the bacteria would go like, there's nothing I can find here that I can convert. And then it'll find a way. There's something that's on board one of those rockets that's like, oh, this whole planet to myself? Perfect. Let me get this started. I know what to do. Yeah.
Mm-hmm. I don't know.
I'm sorry. It was interesting that we're finding it in this way, but we've talked about this before. Like the first life we find on Mars is probably going to be something that we brought there. But this is fun to find. Like the first life we found on an asteroid is from us.
It's ours. That was with all of the best contamination practices.
Yeah.
We tried so hard to avoid it. So I think the next question, though, is as we're looking at these samples, right, is now they're going to have to do more than just determine where it's from. Mm-hmm. Is there life? It's when did it get there? That's the next question.
Yeah.
Before or after?
Yeah.
I think it's an interesting question. When in the chain of events did contamination take hold?
I think it's also a fun story for anybody who's tried to do aseptic technique and had something contaminated. Look, Here is every resource possible being thrown at it by NASA, and they couldn't
JAXA, everyone.
or JAXA, sorry.
Yeah.
And they couldn't keep it completely sterile. So you're all right if you've had a little something extra.
Now it's just understanding what's the noise, right? Signal to noise. This is the differentiation. I know I'm always thinking about signal to noise when I'm, wandering around playing Pokemon Go. Blair.
Yes. So
that's it. We have more questions about Pokemon Go before Blair starts.
Pokemon.
Pokemon.
Pokemon Go. So I do not play Pokemon Go, but I can tell you that I know people that do. I never did. I've never played a Pokemon game until like a month ago. I started playing one of the original ones on an emulator on my phone. But anyway, I never played Pokemon Go. I knew lots of people who did. I would watch them kind of walking around trying to catch Pokemon. But it was this, for those of you who haven't played, you've probably seen somebody playing. They look like a zombie walking around looking at their phone, but basically it's a VR experience. So you see Pokemon through your phone's camera in the real environment, and then you have to like throw a ball at them to catch them or whatever. It's and then there's certain areas
where catch them all.
Rare Pokemon show up, and so people are moving through the real environment and catching Pokemon through this VR experience. Now, this game is provided by this company called Niantic, and they last week revealed that for the last, almost 10 years, I want to say, maybe 8 years, however long Pokemon Go has been around, they have been using data from people playing the game to create an AI model for navigating the physical world. So the way it works, they have a visual positioning system. So they use a single image from a phone to determine position and orientation and build a 3D map. And this is based on people scanning the location through their phone and then capturing images. And so they're calling it...
Breaking up a little bit. Say that again.
Sorry. I scrolled the page, heaven forbid. So by taking pictures and using the VR filter on the game, they are scanning their natural environment, and they are creating a visual positioning system, which uses what they are calling a large geospatial model, or LGM, which is a parallel they draw to a large language model, which is what ChatGPT uses. So language models process text. In this case, this model processes physical space using geolocated images collected through the app. And so they have over 10 million scanned locations, with users capturing about 1 million new scans weekly through the Pokemon Go Scaniverse, is what they're calling it. And so the interesting thing I think about this is, I'm sure it's in the terms of service.
Mm-hmm.
People probably don't know that. Do they care? Who knows? But I think it's interesting that they found a way to, like, if they had an area that was a blind spot in their geospatial model, they would send a rare Pokemon to that space. And then people would crowd to that space to scan that space for them, which is really smart. Also, think about 8 to 10 years ago, what we're now calling AI looked like, what these models, these learning models looked like, how it was if at all. This is like a long game that they were thinking way far in the future when they started collecting this information.
I find this fascinating because this is, we know that Google has been collecting this spatial data for a long time with its Google mapping for streets, and then that they have cameras set up that do 3D stuff to get into buildings and shops, and that all of this is heading in a very interesting, like, augmented reality direction. But I think you're right about the long game here. But what I find fascinating is that collecting. It's not people collecting Pokemon Go. It's Niantic collecting people to locations to use that three-dimensional data, because you have people, like you said, coming from all directions to snap a picture, to scan a Pokemon.
I think
Right.
it's brilliant.
And I brought this up to Brian, my husband, who used to play this. He actually was playing Pokemon Go when we first met.
He did.
Yeah, and he loves Pokemon, and so he really enjoyed the game. But when I was telling him about this, he brought something up really interesting that I did not see in any of the coverage of this story, which is that Niantic originally had a game, and I think it's called Ingress, but it was basically a mapping game that flopped. It was a huge flop. And then they basically put a skin on it to turn it into Pokemon Go. It's almost the exact same code. And so they, even before that in 2013, when they released that game, had this in mind. And it didn't work. And so they changed the aesthetics and
they partnered.
Yeah, exactly. Yeah. And then they were able to pivot and still do their essentially data mining through real humans to be able to make their spatial model. So I think it's a really interesting story of, first of all, like the kind of forward thinking of this company and their ability to plan long term to build this huge model, but also a reminder that when you agree to your terms of service, if you're not looking, you could be helping build a model, which isn't a bad thing necessarily. But the fact that you're using a VR system means you're giving this application access to your camera all the time. So they have pictures in your house. They have pictures in your backyard. They have pictures at your friend's house. And is that okay with you? Probably. Who cares, really? But but it's something that if you don't feel comfortable with, you got to pay closer attention to those terms.
Well, so there's an interesting question, and I have no idea. Would these pokey things show up in private places, or would they generate in public places?
Yes, they showed up everywhere. They would
show up in your house. They would show up on the street. Yeah. So
there were controversies about particular Pokemon showing up in certain museums. I don't want to go.
Or like somebody could knock on your door and be like, "I'm so sorry. Can I go in your backyard? I'm missing."
So it was actually a huge problem when I still worked at the zoo, because Pokemon would show up in animal exhibits and people would try to jump fences. And in the park system, people were like walking off cliffs or going into trespassing areas that were not safe to get to Pokemon. And so like Pokemon Go would have to change things, issue apologies, move gyms, or what they're called, where you can like train your Pokemon. So anyway, there was a lot of stops and starts, and that was part of it, was people were going to private property to try to take pictures of Pokemon. Yeah, it was a whole thing.
So I know this was a big deal because one of my supervisors at the place I used to work was obsessed with this.
And that was a problem. No.
And he got he swapped phones with his brother, who is like a high-level IBM computer fix-it tech. But he flies all over the world. And he was going to Japan, so he was like, "Here." They swapped phones. So he was like, "Go collect some Pokemon from Japan for me." And so he's got like this IBM exec type, like going around collecting Pokemon for his brother. Like people were obsessed with this. And I kind of remember hearing some of the news stories of like people wandering into traffic, or wandering around parking lots in the middle of the night and all kinds of weird stuff. But I do think it's brilliant that this may have all been a data collecting exercise that just had to hang the right carrot out in front of the humans to get them to go do a lot of work for them.
Yeah, and this press release that they have here for their visual positioning system, and people can position themselves in the world with centimeter level accuracy.
Wow.
So digital content can be placed, so augmented reality, digital content can be placed against the physical environment precisely and realistically. It's persistent, so the content can stay there after you've left to be shared with others. And these user scans, from a pedestrian perspective, they say they've 10 million scanned locations around the world. Over 1 million are available for use in the VPS service. And they're building more and more all the time.
Mhm.
Yeah. And also the neural models are out there that they've have some research papers from Niantic Labs. ACE out of 2023 and ACE 0 this year.
I know my hometown is fully mapped. We have a nerd university.
Yeah.
Oopsie David, love you. But it was very popular amongst all of the students, but especially the science and engineering crowd. Kids were like really, really dedicated to getting every Pokemon in town.
I just want to know where this can go, beyond just allowing an open data set of the visual positioning system as opposed to a GPS, this VPS, right? The visual positioning system in combination with GPS and in combination with other databases of information. Can this be used with iNaturalist? Can it be used for crowdsourced research? That depend on observers to track where and when they've seen things? Can it be used? Is this only going to be used for the benefit of Niantic? And selling stuff, or can this be accessed and used by the academic community to allow for an even greater richness of understanding of our world based on people?
So one thing I think it'll do is it could provide a better platform or a similar platform to the Google mapping, right? Here you have one where people have gone away from roads. Most of Google mapping is just driving and pointing the camera in every direction. If you have people wandering down walkways and pathways and through parks and other locations, now you're getting that sort of walkthrough that you can do when you kind of go down the street in those frames of the Google map. With this, you could, and they could put a Pokemon anywhere to fix it if they were missing. You could walk everywhere, through a park, into somebody's backyard, maybe. And I'm curious then too to see how many places used this that Google Maps isn't allowed or doesn't go. I'm thinking specifically like, I want to wander around this small village in China, and there's no Google Map that'll drop the fella down there to walk the streets. Maybe they were really into Pokemon, and maybe we have those maps of everywhere that we wouldn't expect. We might not. We might have mapping of more places than than Google Maps has been able to access.
Yeah, and from the map that is available that they are sharing from Niantic at this point in time, this is definitely biased towards where people are. So all the mapping that they're doing
to where Western people are
is right. Yeah. But Japanese, we have Southeast Asia, not China, not Russia. But the coasts of Australia, we've got the coasts of South America, most of Western Europe and North America, with the central, the mountain and central time zones, kind of less less represented in the North American. Interestingly, Canada, not really. I guess Southern Canada.
Yeah, you gotta figure that most of Canada
not been out in the tundra. Yeah,
even Canadians don't want to live there.
I know. Yeah,
and
Iceland,
the ones that
not
do,
so much Greenland.
the ones that are out there, doing the survival homesteads in the far, far north, maybe not into Pokemon. It might be a non-crossover situation.
That's fascinating. That's super fascinating. This is so many levels of interest, from just data to potential science to artificial intelligence to computer games to just society and the implications for understanding ourselves that come out of this. I am excited. Go on, catch them all, everyone. Give us more information about the
Amazingly
world.
so well integrated into global information system when they're doing all of this sort of mapping. If they want, forget sending out a survey team paid for by your university or by the government or by whoever pays for the survey team to go out and look. Just throw the ultra rare Pokemon out in the middle of this desert, and guess what? You'll have all the maps you need.
People will wander out there, no matter whether they have food or water or doctors. Who cares? Speaking of places that probably don't have a lot of Pokemon, but they might. I don't know. Antarctica. Antarctica is a place where there probably haven't been many Pokemon found over at the entire continent. But we have research labs out there. So yes, Pokemon have been discovered at Asmundsen and other of our research bases. But what about the ice cube detector that's near this Amundsen Scott base? This is the South Pole, the Ice Cube South Pole Neutrino Observatory. They've been collecting data on these weakly interacting particles for a very long time. Neutrinos, they come in lots of interesting flavors, right? Why are we interested in neutrinos? Well, once again, they are high energy particles that give us information about the universe. They have mass, and the various flavors interact in specific ways that have been shown by the standard model of physics, and they have gravitational interactions, weak force. Anyway, the sterile neutrino is a hypothesized fourth flavor of neutrino that would have a little bit of mass, but doesn't interact, unlike the other neutrinos, through any kind of force hypothesized for this through the standard model, but only through gravitational interactions. So if they existed, these sterile, really gravitationally interacting particles, if they interacted, it would get rid of some anomalies in experiments. It could tell us about cold dark matter, warm dark matter, etc. So anyway, the Ice Cube collaboration has, along with some other groups out there, have been trying to see if there is a sterile flavor out there, if there is indeed a gravitationally existing, interacting neutrino. But the answer is no. They just looked at 10.7 years of data at the Ice Cube Observatory, and they didn't find anything. This is an update to past data in that the researchers are basically adding new data to their data set, trying to figure out if this thing is out there, get their significance better so that they have gotten rid of other options or possibilities to explain their results. But basically, the distributions of the energy that they are detecting at the Ice Cube Observatory Array is that, no, there just does not seem to be a sterile neutrino. This is at a level of about 3% significance. So we're getting higher and higher significance with the more data that they are collecting. But this also falls in line with the Mini Boon, Minos, CDHS, CCFR, and Super Kamiokande experiments that have also been looking for the sterile neutrino as well. Nobody is finding a sterile neutrino that's tiny, high energy, and only interacts based on gravity. It's not there. Nope. It's not there. So we can't use a sterile neutrino as far as things work now for the math as a fudge factor in figuring out what's going on in the universe. I love this story though, because what it does is it digs into the kind of the fudge factors that we've talked about. It's like, okay, if we had this particle, it would explain this thing. And so they've been looking for this particle and they're like, okay, well, now we can't. So with a higher probability, we know it doesn't exist. So now they have to come up with other hypotheses to explain their anomalies. Is there cold dark matter? Is there warm dark matter? What? Anyway, it's cold at the South Pole. Not very many Pokemons there.
Yeah. So I have already said too much about dark matter. Or I think it's one on my show. I'm not going to go on.
It's not necessary.
But yeah, they're trying to. Oh gosh, the effort to find particles responsible for everything is it's going to if it's not particles, if it's we're talking about emergent behavior's the emergent things that are emerging from the actual structure of space-time and the fabric of the universe and how that operates, then you can create a particle, you can invent a particle that can facilitate some of these things, but now we've looked for it and it's not there, and you have to come back to the theoretical drawing board.
I mean,
but
there are other.
Don't hit me with another particle.
But no, they have. I mean, this is one of the possibilities, right? So it's like there are all sorts of theoretical ideas out there. The sterile neutrino is one that would explain some anomalies, and so they're like, okay, let's look for it, and they have not found it, and the probability of finding it is decreasing. So, what this means is that other flavors of neutrinos very likely do not turn into sterile neutrinos. That sterile neutrinos are not probably coming from space to hit to shoot through our planet. That sterile neutrinos are probably not being are probably not decay particles from cosmic ray interactions with our planet. And so the probability of the sterile neutrinos being out there further away is less and less and less and less. And so, there are other things that they're looking at concomitantly, but it's it's just multiple. It's like, let's look at this. It's the null hypothesis, right? You want to, we don't prove anything in science. It's disprove ideas, right?
Huge believer.
And there's always multiple ideas out there.
And it is probably the coolest looking research center I've ever seen.
I know. I want to go to really Ice Cube.
They got a nice.
I don't know why it's red at the bottom there. Is that hell? I don't.
I think it's
cool lighting for a photo.
I think it's a time exposure. If it's a real photograph, if it's not just all hacked together,
it is a real photograph. It would be a
time exposure, because that's how they got some sort of resolution on the Milky Way, and that's why
and somebody put a red light out.
It is the sky on fire.
They put a red light out because they're like, hey, that's the complementary color of the green aurora that we have.
Like, what is it illuminating? I can't even tell.
It's probably some like door that opens once in a while and has a flash, a light that like flashes when the doors open, or
probably
like something silly like that. But then, because that picture was taken over, 12 hours, like.
It couldn't have been over 12 hours. The Milky Way
could have
is
been
too
because
clear.
of where it is.
Yeah.
Because of where it is.
Yeah. Anyway, I love these on-planet detectors that are these massive things. So Namibia, 1,800 meters above sea level, and Ice Cube is 1,000 meters to 2,000 meters below sea level, or the top of the ice level. They're just different places that we are trying to get clear signals of the energy that our Earth is swimming in, right?
Mm-hmm.
Yeah. Tell me about health now, Justin. Take us on a
What
different
this is about
adventure.
is also about getting better data.
Sweet.
So this is it's not a study so much. It's a little bit of his people sharing their research, as well as coming together for a kind of a research letter type thing, talking about, or a perspectives letter, talking about what we should be working on next. There's a bunch of institutions: University of California, Los Angeles, University of Washington, Seattle, a whole bunch of others got together and collaborated on this. But what they're talking about is creating a embedded recording device, an embedded cellular recording device that you could place in different tissues. And it would basically be an engineered cell that you would incorporate into your own tissues that would be recording interactions that it has in its local environment. It could be recording its developmental history through generational replications. It could be identifying all sorts of things. But the idea is these cells would be designed so that they basically express an RNA or a protein that then you could collect in a urine or blood sample. And within these specific RNA or proteins that these cells produce, you would have an encoded history of that cell's experience within the body. And so you could use this. You could use these to detect if cancer is beginning. You could use it to detect if they're seeing signals of a specific disease based on basically the life's experience of the cell. And they've got a couple of really interesting suggestions on sort of where we should start with this. There are some that have already been sort of created. We've had modified gut microbiota that were then able to report on sort of their life history within the gut. But yeah, you would want to, some of the suggestions, you want to use a patient's own cells and engineer them so that there's no rejection or anything like that. You could also have them set up with sort of a circuit that if something was going wrong, you could kill. You could have like a kill switch built into them. It's sort of interesting too, because you would have they would be heritable. So they would be doing transcription, replication generationally and maintaining that, not necessarily maintaining the information generationally, but maintaining the ability to send out proteins that are giving these updates. You wouldn't need to locate the tissue again, right? You wouldn't need to go in and sample that tissue to find out what history it's recorded, because all of that data, all that output is getting put out
there,
getting kicked out, and you can just sample it like you would do for a normal screening. When you do a normal screening, though, you can, with some exceptions, you don't necessarily know where it's coming from. We've recognized that there's a signal in your blood that suggests there might be cancer somewhere, right, in the body, like somewhere. We don't know where. But if you have 5 locations where these cells are placed, now you know that if this cell is reporting it, that there's an issue in this area, and you can compare it. Is this a whole system-wide thing? Is this tissue-specific? Because if you have 5 different cells reporting different areas, maybe different, some organs in the body, and they're reporting, and some are encountering this phenomenon and others are not, then you begin to be able to localize where the problem is. But I thought it was interesting. We were talking about lab on a chip a little while ago.
Yeah,
This is like, why put all of that outside of the body where it's just going to give you something happened in the body. We don't know where. We don't know when. We don't know how long it's been going on. We don't know what the progression of those specific cells are. And here you could have you could be sort of putting sensors within the body that are cellular, that grow and probably produce, probably don't do a normal function. They'd just be sentinel cells, basically just there to report to you. That's all they do.
I think this is interesting because what it's doing is it is taking advantage of the body's own kind of natural reporting system to a certain degree, right? Where it's just learning how to harness the information that's there so that we can use it as a sentinel, because like the body's doing whatever it's doing, paying attention to its stuff, right? But yeah, so this is important to know. Okay, how is blood sugar? How is when are these genes activated? When is this particular response happening and why? We've talked about also the lab on the chip. It's also you go in once a year maybe and have have blood test or whatever to kind of monitor progress of stuff. And if you have this additional information, it would give such a richer understanding of aging, of health, right?
Mm-hmm.
Yeah.
Breast, cervix, colon,
Heart,
have
brain,
little
organ,
groupings of sentinel cells and
Mm-hmm.
typical disease hotspots, like cancer hotspots. Especially if there's a family history or something like this, and then you're getting that information well ahead of any sort of clinical observational presentation of the disease. Right now, you're getting it. Now you're getting it at a time point where intervention might be able to completely resolve the issue before disease and illness really have an opportunity.
Right. So being able to see exactly how things are changing. So this is just a model that they've set up, and they haven't actually done it yet.
So they haven't done it. So what they have is, so they have, there's a system that has been built by I think it's specifically by these groups. One is the courier system, where cells were engineered to export RNA barcodes that were able to, send information out, basically engineered to be able to send basic collectible data out. There's the one I was talking about with where they did the engineered bacteria that were, able to be collected through fecal samples that were, sort of telling about had recorded their nutrient availability, acid, stress, inflammation, interactions with other microbes. What signals that they had encountered were captured within there. So they have a couple of the pieces.
Yeah.
But what they're asking, they're sort of the call to action of this perspective is like, look how awesome this idea would be. And yeah, there's all of the challenges of getting there,
actually making it happen. But here's a roadmap.
Here's a moonshot, right? Yeah. That we should be focusing on.
And also it gives, like I said, the roadmap, it gives direction. How do you actually get there? And this is putting all those pieces together. So now people can go, okay, these are the things that are needed, and different groups can work on different parts of the puzzle.
Yeah, I've never heard of this idea before, and I think it's pretty brilliant.
I think it's fantastic. Interesting. Okay, everybody, go. Make it happen.
Yeah, go do it. Come on, science.
Do it.
We want to talk about the results next week.
Go science. Woohoo. Have you ever wondered where your peaches come from?
No.
Thank you for answering that question. Okay.
I assume everything would be from somewhere in California. I never imagined that my produce in California comes from out of state.
Right.
I mean, all the produce
avocado
is not as it once was, right? Like all of our produce has been somehow bred and engineered to look and taste different for factory farming than than it
Yes.
originally was, right?
But I don't know
Indeed.
where peaches were. Peaches must be American. I don't see people going peach crazy. Like people in Europe aren't always like, "Oh, the peaches in Spain are amazing."
Yeah,
Right?
where is stone
must-
fruit from? That's a great question because it's probably like the peach, nectarine, plums. They're probably all related, right?
Avocados, even, right?
All right. So yes, I love all your ideation over here. You're working through the problem. Yes. So those of us who come from the West Coast in California, we know the Central Valley has lots of peach tree groves, lots of stone fruit, and you could say, "Hey, California, whatever." That's not where they came from. Nope, peaches not from California. You're maybe in the South, and you go, "Oh, the Georgia peach." That's the state fruit, right? So, of Georgia. So maybe the South, whatever. Yeah, nope, not where the peach is originally from in North America. According to a study that is just out,
Ohio?
open access in Nature Communications.
It's Ohio, isn't it?
No, you're just going to start guessing states? This
No,
is gonna be all day.
what is that? What is that song? It's like from a total Midwestern
Millions
band. They
of
might be giants.
peaches,
And
peaches
I
for
think they're
free.
from Ohio. I just assumed. Iowa, Illinois, one of those Midwestern things. Sorry.
Okay. Peaches were harvested very early in China, domesticated in China, and then spread across Eurasia via the Silk Road, and then into the Americas as late as the 15th and 16th century via, you said Spain, ding, 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 Spanish colonization. And peaches on this TWISgiving week, it's interesting to note, they became the first Afro-Eurasian domesticate, that's plant or animal, to be adopted by and spread through indigenous communities. So, how did the peach spread across North America? Well, 15th, 16th century, in the early 1500s, it was brought to the Americas by Spanish colonizers. Those Spanish colonizers brought the peaches, and the peaches didn't spread immediately. It actually took about 100 years or so for the indigenous communities to kind of go, these peaches, interesting. Maybe I like them. I don't know. So the Spanish brought them and started planting peach pits and started cultivating the peaches. Indigenous communities eventually grabbed onto this. So the Georgia peach, that actually is very close to where the original peaches came into the United States. The United States, originally, these Spaniards came into the area in Alabama, Georgia, and traded with members of the Muskogee Creek Nation, who currently grow peaches as a heritage or heirloom crop, and they care for the trees, and it's culturally important to them now. But it was about 1620, which is around the time that we date our first Thanksgiving taking place. It was about 1620 when there were the first evidences of or datings of peaches being spread across North America by indigenous communities. So these researchers were like, hey, let's look at old peach pits that we've dug up in archaeological places, and we're going to do some carbon dating there. We're going to look at other chronological modeling that we can do, and we'll try and figure out, where and when the peaches came into the United States, into North America, and how they moved through the country. And really, it took about, like I said, it was from the 1500s until about 1600s that the peach was picked up and started moving through North America, thanks to the networks of indigenous people across the United States. So,
wow, that's awesome.
Yeah, kind of neat that they're able to do chronological dating, be able to determine when the first peaches were detected thanks to Spanish settlements, and then be able to start dating from there, where different evidence shows up through the years into, when peaches kind of became normalized across the United States. Yeah.
That's cool that, like, basically because it's a stone fruit, it was easier to transport, I would assume, across the ocean, but also easier to date in this research. So that's the kind of the nature of the stone fruit made it the perfect candidate for this research.
Yeah, and we know if you ever break into the stone fruit at the center of a peach or a nectarine or one of these fruits,
I've never tried.
it's like a walnut, right? It's a very hard stone at the center, and the seed itself is within that. And so very often like walnut shells and other actual nuts, the husks, the husk of the stone fruit itself, that seed in the center, is so hard that it is often preserved, and they were using it where the archaeologists actually found a lot of their original bits of the stone fruit were at the base, kind of like gravel of posts for houses that had been foundational pilings that were used as structures to build structures in these settlements. So they used the broken stone fruits and nut shells and everything, and they put their pilings for structures in them, and so they're able to use that. Like you said, they preserve for a much longer period of time. Yeah, but so it was much later, pre-1600, when the peaches came in, and it was after 1650 to 1675 that peaches, thanks to indigenous communities, actually began to spread across the entire United States and North America. Indigenous communities! I'm going to have to make a peach pie for Thanksgiving this year.
Very cool. Yeah, I'd never, no idea. I'm here in Europe. I don't think I've seen a peach in Europe yet. Maybe I just don't go to the right places.
You need to be in Spain and not Denmark.
Yeah.
Lots of peach groves in Denmark. No, there are not.
None.
Hey, everybody, this is This Week in Science. Thank you so much for joining us on this TWISgiving episode. We're so glad that you're here. And it is my pleasure to invite you to TWIS.org, where you can find more information about the show, each episode, links to our stories so that you can dig deeper into each of the stories, into the original source material. Additionally, you can subscribe to our podcast, subscribe to our live streaming channels, at TWIS.org. And if you are beginning to search for those holiday gifts, you can click on our Patreon or our Zazzle links at TWIS.org to do a little bit of that directed shopping that will support This Week in Science and also get TWISy goodness for the people you care about in your life. I mean, really, if you're going to share anything this holiday season, share the gift of TWIS. Right? Why not? And next week is Giving Tuesday, and your gift to TWIS could very much be to help support us into the new year. So we ask for your support. Click on the Patreon link. We also make PayPal donation available. Please give to TWIS, as your gifts are what really keep this show going. Thank you so much for your support, day in, day out, week in, week out. Little bit of stories of science shared around the world. Thank you. We can't do it without you. All right, as we're going to come back right now, it is a part of the show that we haven't heard from in a little while because Blair has been off gallivanting. No, really, being a mother, having a family, being responsible. It's time for Blair's Animal Corner with Blair.
She loves our
What you got, Blair?
I have a story about Ethiopian wolves, which,
What?
if you're not familiar with them,
I am
they're a
not.
wolf. They're just a wolf species. They're really cool. They're endangered. They're very rare. We actually had a statue of one at the zoo when I worked there, so I learned a bunch about them at the time. They're really amazing animals that aren't well known, but some new research from the Ethiopian Wolf Conservation Program have found Ethiopian wolves doing something really unusual. They found them foraging for the nectar of the Ethiopian red hot poker flower.
They're so cute. They look like a fox wolf.
Yes, absolutely.
They're-
They look like a
they're-
fox wolf.
Yeah, they're adorable. And okay, so this looks like this picture that comes with the study. It looks like this cute fox wolf,
Mhm.
Fantastic Mr. Fox,
Mhm.
is licking a one of those 4th of July ice cream rocket pops.
Yes, yes, absolutely. And so what they discovered was that this particular flower has this nectar in it. And so the nectar is very sweet. And these wolves will go sometimes up to 30 blooms in a single trip, lick these all up and down to get all of the delicious, delicious nectar. And in the process become covered in pollen. And so that's what I assume you can see in this photo. I can't see it very well because of the color blindness, but I think their nose is all yellow.
Their chin
they-
is all yellow. It's
Ye-
like a 2-year-old, except it's the Fantastic Mr. Fox. And they've gotten that orange, that red, yellow, orange rocket pop all over their face, their snout.
There you go. Perfect.
Mhm.
Yes.
Mhm.
So in this process, this means what? Ethiopian wolves pollinate.
Pollinate.
Yes. Yes. So as they see 30
Oh, I
blooms
was
in
gonna say,
one trip,
yum.
yes, they are pollinating. So they
Oh.
are transferring this pollen from flower to flower as they feed. This is what researchers at University of Oxford believe is the first known plant pollinator interaction in a large predator. I would put a caveat on that and say potentially an intentional foraging that results in pollination in a large predator, because obviously there's lots of pollination that happens by animals rubbing up on things and then rubbing up on other things that is incidental, right? It's a
Right,
coincidence.
yeah,
It's just part of their
not a
environment. They're moving
regular
through their
thing.
environment. But yeah, but this is a
This one though is
foraging.
very bee-like. Right? This is-
Exactly. Yes. This is also interesting because wolves are generally speaking carnivores, though the more we learn on this show, these herbivore, carnivore, kind of black and white boxes are gray at best. So they're eating nectar. Otherwise, they're meat eaters. It's kind of unusual, but that's kind of the least of my concerns from this story. I think, my experience working with large carnivores is that they'll eat a lot of things, even if they are supposed to be carnivores, because animals are just trying to survive out there. So they're going to eat whatever food is available that gives them the nutrition that they need. And if there's delicious, sweet, sweet nectar available, why not?
So
yeah.
now I want to know more about the flowers and how much nectar they produce and how often. And you're saying that these foxes come and they get all of the nectar. They lick the flowers up and down, get the nectar, they get the pollen, and then they're probably going to another flower, right? And the transfer happens and everything.
Yes.
We know that hummingbirds, which are pollinators and nectivorous, that hummingbirds have a memory for different flowers that they have visited, so they know not to waste energy going back to a depleted flower. And so what I want to know is, how much do these flowers impact the energy metabolism of these foxes, and if it is a big energy. Oh, wolves, sorry. I want it to be a fox.
Foxes, I know.
Of these wolves, then how impactful is that to their behavior? And do they have a very specific memory concentration game of
Right.
which flowers they've visited? Which flower are they paying attention to what other wolves have visited flowers? And, like
Right.
I go visit the
Yes.
flower, or are they just snarfling all over the place and going, this one sounds good?
Lots of really good questions. I have some answers. But also I think the other question that I could tack onto that list of questions is this flower creating nectar for the wolves? Or is this
Right.
something that a lot of different animals do? And it's just like, hey, come on,
Yes.
come all, enjoy the sweet nectar, right? And so the
Yeah.
wolves are just taking advantage of something that is created for a large host of animals. Or is this a
Yeah,
very
so different from like the honey creepers and their beak specializations and the flowers that they visit. What's happening?
right?
Yeah,
So
evolutionarily, biome evolutionarily,
Mhm.
how long have they persisted together, and what would have been, did they displace another carnivore, large mammal that was engaging in a similar activity previously?
So, to my knowledge, I didn't brush up on my Ethiopian wolf facts before this show, and I should have. But to my knowledge, they are an animal that has been around for a very long time in that space. The problem is actually the opposite, that they are endangered, and so their population is decreasing. So my question would be, is that impacting the flower's ability to pollinate and propagate, right?
Mhm.
The Ethiopian wolf is really well established. It's been there a very long time. It's not a newly introduced animal. We don't know how long they've been doing this behavior. But one of the things that I think is really funny in this story is that Professor Claudio Solero, who's the founder of this conservation program, and he's based at University of Oxford, originally saw this behavior by watching humans. He saw children of shepherds in the Bale Mountains licking the flowers. He tasted it, was like, wow, that's really sweet. And later saw the wolves doing the same thing. So, also
People figured it out because of the wolf.
Right, yes. Could be the opposite, who knows? But also the wolves seem to pass this information on to each other as well. So they will bring juveniles with them. Older wolves will bring juveniles with them to the flowers to show them kind of like, this is the good stuff, right? So there's some generational knowledge being passed down as well. So obviously there's a lot still to learn. Go ahead.
And then the one more data point that I would then want to find is, I would want to find a very old version of this plant that we could sequence. I don't know how possible it is to get a 6,000, 5,000 year old version, and see if they've become sweeter. See if the sweeter ones have been pollinated more because of this interaction with the wolf, a prolonged period of time, right? Because this could become a form of cultivation, that because the wolves
are like ants,
are licking more on the sweeter ones, that the sweeter variety are the ones that are getting propagated more. And this is in a way how humans created all sorts of food sources that we now take for granted, that didn't look anything like what they look, the food source that they are today. 10,000 years ago. It's because there was a whole lot of natural selection and human agriculture that was involved, and this could be wolf agriculture for the first time, which would be really fascinating to see if they've impacted the evolution of the plants by doing this.
I'm sure they have. Yeah, but that would be the interesting question.
Yeah. So there you go. Large carnivore pollinating, enjoying some sweet nectar.
You too could be a pollinator. No, maybe.
Yes. I guarantee all of us have at least accidentally pollinated.
Sure.
Moving on from the terrestrial to the aquatic, a multivitamin helps save coral reefs. We've talked a lot on the show about vitamins and how at least with human vitamins, they're like not very well regulated. You don't know if it's actually doing anything, how much is even in there. Well, So we're going to use the word vitamin here kind of loosely, and I'll explain specifically what this story is about. But this is research out of Woods Hole Oceanographic Institution, and they have a lab where they have been testing nutrient-infused tiles to grow coral on, to determine whether it would boost the immune system of corals that grow on those tiles, which of course would help them withstand stressors like warming ocean temperatures, extreme climate events like hurricanes, and then also the result of those, which is often coral bleaching, which is when they expel their zooxanthellae, the kind of symbiotic relationship that they have with it that is the reason that they have nutrients in their bodies. Coral can't get their own nutrients, so the zooxanthellae do that. Anyway, long story, getting way in the weeds here, but basically a lot of coral can't survive bleaching. It's too stressful of an event. And so the reason coral reefs are being hit so hard by climate change is that it's just too many things happening at once. It's warm, the water's acidic, nutrients are less, there's hurricanes happening that increase water depths, which change all sorts of other factors. And it's just everything happening at once to the point where the coral gets so stressed out, they bleach, they can't get their zooxanthellae back, and they are toast. They die. So does giving them, like we take zinc when we feel like we're getting a cold to boost our immune system, right? Can we have them grown on tiles that have manganese, zinc, and iron in them that boost their immune system and help them recover from stressors?
And because they're tiles, it's not just seeding the local water with extra nutrients. It's actually a mineral upon that the organism has to like use with chemical interactions to actually make use of those minerals, right?
Yes.
Yeah,
so
I'm just wondering, because we've had the studies that suggest that, like, we can put bacteria in the water or yogurt or whatever for coastal yogurt.
Yeah. So, I mean, part of the problem here, and the reason that there's a symbiotic relationship between the coral and the zooxanthellae, is that coral reefs, what do you picture when you think of a coral reef? You picture this pristine ocean, very clear and beautiful. Clear waters are actually vacant of nutrients. Disgusting, muddy, silty, thick water, like found in like the San Francisco Bay, is full of nutrients, full of oxygen, full of everything animals need and plants need to grow and survive. And so coral reefs are actually the deserts of the ocean. They are void of all of the things that plants and animals need to survive. And so coral reefs have developed very specific relationships with their photosynthetic algae to create nutrients for them, because there's not enough nutrients in the water. Remember, coral are little, like, basically sea anemones that are grabbing stuff from the ocean to try to eat, but there's nothing there. So the algae is what supplements that. They also have these beautiful reefs that they develop to attract fish to poop all over them. Nutrients, protect
nutrient
them.
cycling.
So they are, yes. So they are creating their own little mini rainforest amidst what is essentially a desert, right? And so if sprinkling things into the general water table around a coral reef is not going to be very effective, because that water doesn't keep things in it. The reason the San Francisco Bay is so silty and thick and full of nutrients is that it's cold and it's nutrient dense and it holds on to those things. Coral reefs are warmer. They don't hold as much nutrients or oxygen. And it's just the whole water dynamics are different. And so if you put general nutrients into the water, they would not stay. And there are all these kind of other issues, right? So putting it in the tile means you're infusing it, like you talked about, like straight into the coral. The coral can take it up as it grows. And so this is a really cool idea, but there's several steps to this process. This was done in the lab at WHOI. And so this
Woods Hole Oceanographic Institute.
Yes, sorry.
WHOI.
That is what they call it internally. And I went through some trainings with some WHOI scientists, and so I sometimes say WHOI. Apologies.
Sometimes you're talking WHOI.
Yeah,
Is
exactly.
that right? This is really good research. Yeah, it's like a bunch of hooey.
Yeah, exactly. Yes.
Oh my gosh.
I got to go to Hui and see their cores. It was amazing. Anyway,
Oh, neat.
the rapidly warming water, coupled with extreme weather events like hurricane season, means they could only do this research in the lab. They wanted to do it in the wild, in the field, but they were not able to do that because of all of these other kind of circumstances going on at the time. So they had to work with preliminary lab data first. So they did a year of lab experiments. They showed that the corals had early benefits from the multivitamins and that they were generally more resistant and resilient to heat stress. But so the next step now is to bring this into the field. They want to embed these nutrient-infused tiles into the concrete structure of an artificial reef that's about 20 square meters that's designed by the University of Virgin Islands. This structure will be the foundation to plant juvenile corals and for coral larvae to settle on. And there will be a section with tiles and there will be a section without tiles. And they will be able to see if there is a difference. Because the other piece here is, yes, in the lab, when you only have these very specific species that they are testing with the tiles, they benefit. Do other species benefit? Do invasive species benefit from these tiles? Do they benefit more than the native species and actually cause further problems and further stress because the invasive species are growing insane? Do they only help one specific type of coral? These are all really important questions. So they need to be able to ensure that the artificial reef looks and sounds similar to a natural reef. They need all of the same benefits of the reef there, beyond just like, yes, it helped this set group of species that we tried in the lab.
I'm going to play the uninformed science skeptic.
Yes.
And say, why do we even care about coral reefs, Blair? Why do we care that we have coral reefs?
You just want me to say the coral reefs are the lungs of the ocean and they create more than half of the oxygen that we breathe every day.
Is that what we're breathing? It's the oxygen and they're making half of it? Oh, that seems really important when you put it that way. Oh, gosh.
Yeah, it's all those photosynthetic algae that I was talking about. So coral reefs are pretty gosh darn important. I love the idea of these vitamin infused tiles, but this has to be done in conjunction, of course, I would be remiss to not say, has to be done in conjunction with efforts to reduce the impacts of climate change and ocean acidification.
Yes,
Yeah,
this isn't going
to reduce our carbon
dioxide. The other thing.
Yeah. This isn't fixing any problems, but yes, and
help the current stressors, reduce future stressors. Must do both.
The thing that I find it that you brought up here that I was wondering about, this is artificial reefs are not the same as just the usual surfaces that the coral grow on. We are putting places to help rebuild areas that have died out, or to build in areas and help coral populate new areas to help them spread. And so being able to provide a better foundation for coral reefs is beneficial, right? So these are conservation and remediation efforts for the dying coral reefs of our planet. Not just keeping the ones that are already there healthy. It's how do we make better foundations for the reefs that we build.
I don't actually know because I've not ever looked into this. Could we survive on half of the oxygen in the atmosphere that we have?
No.
So
I mean, there would be problems.
Are we saying that if the coral reefs all died out, that it would mean that we would be next? Or,
if all the coral reefs died out, there would be a much bigger problem in the oceans. I mean, it would go. There would be a massive decrease in a lot of stuff.
It seems important then.
It is kind of important.
So everybody stop taking your vitamins because they're needed elsewhere.
But instead of vitamins, should we take cannabis?
That's completely up to you. It's a personal choice, as far as I'm concerned.
Justin?
Oh no, I have a story about
is
this.
Is that something that we should be concerned about? It is your story. I know.
Yeah. So okay, so disclaimer though, this was a study on youth and adolescence. So this isn't like a grown-up study. However, one of the fears about the reefer, the marijuana, the cannabis has been that it causes psychosis in youth. Like, oh, if you do this, you'll end up with a high risk of getting psychosis of some sort as a result of it. And there's actually three separate theories as to why there seems to be a real attraction to cannabis in youth. And what that correlation is to psychosis, because there does seem to be a connection. Like that has been presented. So there's the three sort of scenarios are: one is the contributing risk hypothesis. That means that cannabis use actually causes the emergence and progression of psychosis through disruption of what would be the natural, normal neurodevelopmental process during adolescence, when there's lots of changes taking place in the brain. And so this is like, it's gone in and it's disrupted the natural process. Another is that this is an environment, the vulnerability hypothesis. And that one suggests that, well, there's something else, a genetic propensity, something gestational, something early developmental, something in the environment of the individuals that actually puts them at a higher risk of psychosis already, but that also makes them more likely to try cannabis. Right? Like those two things go together and they're just sort of parallel. And then there's the third hypothesis is the self-medication, which is these are individuals who are turning to cannabis as a means to alleviate distressing symptoms associated with something in the psychosis spectrum. And that's why they're engaging in cannabis use. So this is a study, 11,868 adolescents. They were aged 9 to 10 at baseline. That doesn't mean that they were on the cannabis. This is when they started tracking these individuals. And they tracked them through, that early, late childhood into adolescence. What they discovered was that the symptoms of psychosis showed up first. That it was basically the findings showed that there was symptoms of distress from psychosis spectrum symptoms that were already present and were already building before cannabis use was introduced or initiated for the first time. And so it's kind of reinforcing either the self-medication or that vulnerability where if the things that lead to having psychosis that early also lead you to a propensity to try cannabis. But it doesn't rule out causative, but it has no evidence whatsoever in this study of 11,000 plus that supports a causational relationship between cannabis use and then the onset. It was interesting too because they found some sort of similar things with tobacco use and with alcohol use to a lesser extent. Right? But yeah. So all of you who are afraid because you watched
Reefer
one of
Madness.
those after school special
that
TV
egg in the
shows where they
frying
told
pan.
you-
This is your brain.
Right. Now it turns out though, playing Dungeons and Dragons does lead to traumatic psychosis because Tom Hanks played the role of the kid. And so he's the most believable person in any role. And so that one stands. Dungeons and Dragons is directly leads to extreme psychosis and maybe even schizophrenia. It's causal.
That was because of Tom Hanks and the movie.
Because of Tom Hanks.
Because everybody loves and trusts Tom Hanks.
Because we saw it on TV.
The Wizard.
Therefore,
I remember that movie. I just told my son about it this last week, and he
what
looked
movie
at me
is that?
like, I think it's called The Wizard.
This was
and it was
before
from the
you
80s.
were born, but while me and Kiki were teenagers or young adolescents.
It was
there was an after school, I think it was like a 3-part episode, and it starts with a bunch of friends playing like.
I was terrified of role-playing games.
It was
because of this show.
It was supposed to be anti Dungeons and Dragons. Like people were worried about their children playing Dungeons and Dragons.
This is a slippery
This is before video games. The thing to be
Mazes and Monsters.
Mazes and Monsters, maybe.
And the main character was played by Tom Hanks, and he gets lost in the game and can't tell reality from this fictional universe that they created in the role-playing game, and just becomes lost to them all.
Oh, and then he die at the end. Like it was really fear mongering.
I don't know if he died. I feel like he got institutionalized, but maybe I made a happy ending because I tend to do that. I remember things
happy
better
ending
than they actually happened.
Though he is now in regular counseling, it is implied that Robbie will live out the rest of his life trapped in his imaginary world.
Okay, there we go.
Oh, I remember him dying. I couldn't even watch that show.
I actually kept the right ending. You put a wicked spin on it. But it was like the most and then parents were afraid of letting their kids play role-playing games.
And then came video games. Stigma. Yeah, it was the whole thing.
All right.
Okay.
And I think it might have been like Tom Hanks's first big role.
It might have been.
Anyway, so big study doesn't show a causative effect there. Oh no, I must have one more.
You do have one more.
Let's
stick to the show.
I like the way that's cut into the Gibraltar cliff face overlooking the Albordian Sea. A cave opening leads back in time to one of the earliest manufacturing sites on the planet. A Neanderthal-built tar distillation oven hidden for 65,000 years. So this is a really fascinating study. This is what they have discovered here is within this cave, basically a pit that they can tell was used to manufacture tar from this specific type of leaf. And they can tell that this was, when things were put into this, they were covered, and it was the fire that was started in there was then burning at a low heat because it was deprived of some of the oxygen, which facilitated this conversion of this plant into a sticky tar that they could then use to fix things. They could use it as an adhesive to put on spear points or, just connect whatever tools they needed to connect to whatever else. What's fascinating is that this is the first time we found a manufacturing site. We already had back engineered that they do this underground with low oxygen because the tar, the only way you could facilitate the chemical result of the way that tar is formed is by doing these slow, low oxygen burns underground. Okay. The study that I'm recalling that I think covered on this show probably a year ago or so, where they had done the archaeological experiment where they had done above ground burning of birchwood and below ground and then covered below ground and all of these experiments. The only way they got to the result of the specific tar the Neanderthals were using was by making this pit and covering it and having this controlled fire. The amazing thing to me, though, is that study was in Germany. This is in Gibraltar. And if you don't know where Gibraltar is, I don't blame you. Nobody's ever been there. It's this little rock outcropping at the bottom of Spain.
Yeah.
that's actually part of the UK.
Mm-hmm.
Right. And so
Off the Iberian Peninsula.
the Iberian Peninsula. And so this is a very long distance away, and it's a different material. This is a plant leaf material versus a tree material from the German site. But what this is showing is it's the same manufacturing technique. It's the same covered low oxygen burn method that's being used. To me, that's extremely fascinating because that tells you that this is knowledge that is captured, that is transmittable, that is maintained for a complex manufacturing process that is creating a synthetic material. The world's first synthetic material is this adhesive tar that has to be, specifically modified with applied heat under a specific condition. So, yeah, I find this very fascinating.
So was this then likely to have been spread from population to population by nomadic Neanderthals? Was this Germany to Gibraltar? That is, it's all within the range of the Neanderthals moving around, but
so we assume
so
moving
it's learned
around.
and taught and shared. Yeah.
So here's the thing: we assume moving around.
Yeah.
We don't know how moving aroundy they were. Right? We know that they lived in pretty small groups. At least that's what our current understanding is. But if this cave with this pit, we haven't seen a lot of these elsewhere. So a lot of the technique could just be digging a hole in the ground and doing it. I think we're going to be looking for this a little bit more closely going forward for something similar to this. Mm-hmm. But they could be here making adhesive for Southern Iberia. Okay. This might be
this is the manufacturing plant.
Yeah, it might not be as widespread knowledge, right? This might be hey, let's collect a bunch of these leaves this season, and then we'll take them down to that cave in Gibraltar. And this cave is also, this is directly overlooking the sea. But back in Neanderthal days, 65,000-ish years ago at this site, it would have been actually a decent distance from the sea. Right? It would have been a different environment than it is now. But you can imagine if you've got a really nice big fire pit set up in this controlled environment in this cave, if there are nomadic Neanderthals, if there are other places that may not have cave sites like this, or the knowledge, they might bring them leaves and then trade them for other things to get that sticky tar that they need. We don't know how complex the society was. We are constantly having to update it and realize that there's much more cognitive ability, that there's much more abstract thinking going on with these groups, that they aren't, it's the point of talking about Neanderthals and always having to dismiss the characterization that we've made of them is at this point so ridiculous even. That now we really need to start trying to understand how complex of a society it was. So when I see this manufacturing site as a kind of perhaps unique location that may have been part of a bigger network in the region at the time.
Yeah. It's so interesting because humans populate the earth so densely now. It's just really interesting, though, to try and consider, the much smaller numbers of individuals that were spread across Eurasia.
That we know of.
That we know of, right? But it was not, we're not talking billion people Neanderthal population.
No.
We're talking tens of thousands, hundreds of thousands tops.
But we also don't. Also, consequently, we don't have a whole lot of evidence of them going to war with each other.
No, right? So,
they may have had a tendency of just you know what? The other Neanderthals moved into the cave next door. I think we're going to move 2 caves down. We're just going to keep a little distance.
We can keep, yeah. There was
But this-
plenty of
this
Earth to
site-
go around at that time.
If you think of all of the things you would need to do on a day-to-day basis to survive in the Neanderthal world,
It's good to
spending
have
a lot
help.
of time manufacturing adhesive is a pretty specialized task.
Yeah.
And so you then start to imagine that that could be a role that somebody did, that they didn't go hunting, maybe.
Right.
Like they maybe didn't forage. Maybe they made adhesive. And somebody
Maybe
else, maybe they only fished. And maybe this other group over here, maybe they only did the foraging or the hunting, or maybe did them together. And then brought everything together and shared with each other these things. Yeah.
Bringing everybody together and sharing the things
Always
together. Oh, that is a nice segue into our end of show stories related to Thanksgiving. But before we jump right in there, I'm going to tell one quick story about the brain and reproduction and female fertility. Specifically, not humans. We're going to be talking about little rodents in this particular study. Researchers just published in Neuron, an open access paper on a hypothalamic node for the cyclical control of female sexual rejection. So this isn't when the ladies get rejected. This is when the ladies reject the men. And typically when we talk about reproduction and sexual attraction for mates, that's all that we talk about. We talk about the attraction part. But the rejection is much more common. If you look at any reproductive interactions in the animal kingdom, it's like, no, 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 yes. No, 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 yes. If
Eggs are expensive.
Yeah, eggs are expensive, right? Females are the choosy sex. And it's not just choosing, it's mostly rejecting. So what is it in the brain that's leading to rejection? What is is there a signal that says no, 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 no before saying yes? And so in this particular study, these researchers, they looked at an area that is called the ventromedial hypothalamus, which is a very old region of the brain, and it controls social and sexual behavior across lots of mammalian species, including humans. However, this is not a human-based study, once again. And they know that there's a part of it, the anterior, which is the forward anterior portion of this ventromedial hypothalamus, hasn't really been investigated a lot, specifically with cells that are responsive to progesterone. Progesterone is one of the hormones in the sexual cycle that fluctuates and usually is opposing the impacts of estrogen. So they decided to take a look at these particular cells in the anterior ventromedial hypothalamus, and they found that through these very advanced techniques, fiber photometry, which is able to actually track real-time brain activity through calcium signaling of the neurons, they found that progesterone-sensitive neurons in this area of the brain, in both receptive and non-receptive female mice, were active during interactions with males. These anterior neurons were highly active in non-receptive females, correlating with defensive actions like kicking and boxing, and were far less active in the receptive females, the ones that were saying yes all the time. So the progesterone responsive neurons get activated. They're sensitive to progesterone. They get activated by progesterone that's flowing around, and they are the rejection gatekeepers, say the researchers. First author Basma Hussein, says, when a female is outside her fertile window, these neurons become highly active, prompting rejection. During fertility, activity decreases and allows mating to occur. And they were actually able to turn these neurons on during, and so use like modification of the cells to turn them on and off at like the opposite stages. So females that were in their fertile phase that normally have low progesterone, they turned on these progesterone sensitive cells. And suddenly, once receptive females were like, no, 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 no. And then they turned off the sensitive cells, and the females once again said, yes, 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 yes.
Which I fear was the whole point of the study.
So this is interesting though, because I will say a lot of the conversations around female hormones and the fertile phase is talked about from the male perspective of saying like the males know when the female is in their fertile phase. The males can smell it. The males do the Flehmen response. They will lick urine and test and see, right?
So we are talking about the mice, right?
We're talking about mammals in general. I was thinking about like giraffes, lions, right? Like a lot of animals do this, but you that is the perspective given, is the males figure out when the female is.
It's been a long time since I've been on the dating scene. I just wanted to make sure I wasn't.
So I think this is an interesting change of perspective, that instead of putting the agency on the male animal of knowing when the female is in heat, in this case, the female is choosing to mate or not based on her hormones and whether she is in the fertile phase, which is just a different frame from how it often gets talked about. So I think that's very interesting.
It is, and I think that's one of the reasons I brought it. That this is a different way of looking at the whole system. And the bottom line of the study here is that what they were able to determine is that there seem to be two distinct populations of neurons. These progesterone sensitive neurons that control rejection, and then there's another group that control receptivity. And so together, hormones modulate those two dials, those two controls. And so instead of just having an on-off switch, yes, no, it can become much more nuanced, because you can have more receptivity, but then maybe a little more rejection, or more on the rejection scale, but maybe a little bit of, so it's not just one or the other. There's more nuance to the mating behaviors, and also a way for the brain balancing what they say here. It's a more efficient and robust way for the brain to balance these behaviors, rejection, receptivity, ensuring mating occurs when conception is most likely, while minimizing the risks and costs of mating, like exposure to predators or diseases. There's flexibility to the brain's regulation of sexual behavior. Sex isn't deterministic. And even during the receptive phase, a female might still reject males. So the ability to draw on both these sets of neurons may allow for more nuanced and dynamic behaviors. And then if we look at it in the sense of extrapolation to humans, we can potentially start to understand, if assuming that these populations of neurons are active in the human brain, how these might lead to certain individuals being more interested or less interested in sex at different times of the month or on a regular basis. What are the neural mechanisms and internal states that drive these very, very social, complex social interactions? And we want to know about all this stuff because, thank goodness, it's TWISgiving, and people are going to be coming together and sharing about their lives and talking about all sorts of things. And hopefully there's a lot of gratitude and, thankfulness, because research suggests that gratitude and thankfulness, it's good for our well-being and also our connection, gets the oxytocin flowing for connecting to other individuals. On top of that, when you dress up and you feel pretty, a study out this week suggests that people who feel beautiful are more likely to be nice and more likely to give to other people. So you're more likely to donate or gift to others or be generous to others when you feel pretty. So dress up, be nice, and maybe you'll be nicer at the dinner table. Look at yourself when you get to that Thanksgiving dinner and you're not feeling generous and say, I feel pretty. I feel so pretty.
Or tell cranky people that they look great. Maybe
Right.
it'll turn it around.
Exactly. Yes.
Nice.
If you feel good and somebody's being cranky. Oh, but you look fantastic. Have you seen yourself? You look so good. Maybe it'll just, the giving will get better and better. But what's going to be on our dinner tables this holiday season? In the United States, it is Thanksgiving tomorrow. We are recording our TWISgiving episode one day ahead of this annual holiday that, is contested or contemptuous, controversial for historical reasons.
Only if you think genocide is like a bad reason to have a holiday.
Yeah, sure. But at the same time, we can repurpose old holidays the same way that what is now Christmas was at one point in time a very different religious observation. So we can repurpose things. It's okay.
It's always. I think it's. Correct me if I'm wrong. I think it's always been a pagan holiday.
Pagan, yes, but Christmas is not this. Christmas is Christian, not pagan.
Oh, come on!
It was repurposed.
It's still a pagan holiday.
All the things. Yeah.
Look at it.
Okay, fine.
Let's teach children greed and indulge in overspending consumerism. Yeah, come on.
See, marketing. It has repurposed so much in our lives. So we can appreciate our holiday with the understanding of its difficult origins, but also understand the time together and the experience of the gratitude and sharing can redefine it in new ways that we can make a positive holiday out of something that is historically very fraught. But anyway, there's a great article in Popular Science this week about the turkey, which is the bird of choice for, it's our national bird, right?
Almost.
Anyway.
Almost. And everyone else was like, why would you want this fat, bald-looking bird to be, and then...
They weren't always fat.
Obviously.
They used to. Why have you seen a wild turkey? Wild turkeys are scraggly, like
not the ones in Davis. The ones in Davis are huge. They're huge. I don't know what they're eating, but they're well fed. They're free ranging throughout the town.
Well, they probably escaped from a research facility, but we won't talk about that. They're not capuchin monkeys or rhesus macaques. They're just turkeys, so who cares? Anyway, where did the modern turkey come from? Well, yes, indeed, wild turkey may have been part of the historical meal. There is evidence that the first Thanksgiving in 1621 that we say was our very first thanks, or 1621, 100 religious pilgrims came to the New World, landed in southeastern Massachusetts, which was inhabited by the Wampanoag people. And though there were issues back and forth, this area had also been inhabited there by earlier pilgrims who were not quite as hard they weren't as much of a sticklers for the way that they thought about stuff. But the later pilgrims who came and had the Thanksgiving time, their whole thing was much, much more puritanical. But anyway, the Thanksgiving that they had, it was because they had been having contact for about 100 years. The Wampanoag had been in contact with Europeans since the 1500s. We talked about the peaches earlier along the southern coast. And here, earlier in about 20 or so years earlier, there were interactions with English Bartholomew Gosnold and Martin Pring, and from France, Samuel de Champlain. And the encounters that had happened historically were usually violent and had a lot of kidnapping. But then around this 1620 time, it was like, okay, we're new pilgrims. We're not doing great. We need some food. Our farming isn't doing fantastic.
It was a bunch of zealots and city folk.
Yeah,
who
and they
moved
really
to an untamed country or a wild country or moved to nature for the
Right.
first time. Didn't matter where in the world it was. Had no idea how to survive, and
Yes.
so the native population had to prevent them from starving.
But there had been violence in the interactions prior to this. And so it took a long time to really kind of get things going. And when Plymouth Colony was founded, they formed an alliance, they had a peace treaty with the Wampanoag, where they provided the indigenous people with weaponry and guns, so military technology among things. And so the Wampanoag were able to defend themselves. And then additionally, they shared food and were able to protect and help keep the settlers from dying. And so it was a military alliance, really. And historian Kathleen Duval at UNC Chapel Hill said that the fact that their friendship was a military alliance against the Narragansetts understandably usually isn't included in the kids' version of the Thanksgiving stories. The indigenous tribes, though, taught the English how to fish, plant crops, and gather shellfish. And so the first harvest probably, although it had local fowl, which probably included skinny turkeys, there was probably corn, fish, deer, and other birds. The Wampanoag probably brought corn and meat as well. And additionally, there was probably a lot of shellfish. There was probably a lot of fruits of the sea.
You don't see that in the traditional American Thanksgiving.
No. Additionally, they didn't have a lot. These were early settlers. They didn't have a lot of the comforts of home. So there probably weren't pumpkin pies and those kinds of things. There were probably wild berries and greens from the colonist garden. And they probably didn't have a table, say historians. Duval said that they didn't have a lot of silverware. They didn't have glassware. They didn't have probably they sat on the ground. They probably sat on the ground and ate with their hands and ate a lot of fish and nuts and berries and maybe some meat. So the Thanksgiving of today, that it has changed significantly since that very first feast, and the feast didn't have historical relevance at the time to the people involved. It wasn't necessarily a unique event because the English and the Native Americans regularly held ceremonies and feasts to celebrate the harvest. And so the evidence of this first Thanksgiving is that it's kind of like a mishmash of a bunch of things. And it wasn't until 1863 that President Lincoln gave the Thanksgiving Proclamation that created the holiday. That has actually changed dates over the years, but traditionally takes place in November. So I think it's very important to understand the history of the Thanksgiving feast, the Thanksgiving gathering, the bits and pieces of nuanced information that can be combined when we talk about this holiday and understand its true roots and where it came from. I think, in the bottom of it, it is bringing people together and sharing. Whether that might be militarily or not. But yeah, the turkeys nowadays, the bird on your table is not going to be a skinny bird. But yeah, it's not going to be a skinny bird. It'll be a very large bird. The birds nowadays, they do not allow them to be grown with growth hormones as was done in the past. And the large
no.
bird of today, similar to what you were talking about earlier, Justin, with natural selection, the birds of today have been achieved through selection. So the big breast muscles that are really not great for flight, and the birds now, they often can't fly, have problems standing up on their own, often actually are so unbalanced that they break their own bones. So the bird of today, even though it says it's organic, it's not going to have growth hormones or antibiotics, but it is not necessarily, it's definitely not natural. These birds have to be artificially inseminated. They are, their reproduction is not natural in any way because they're too big to be able to actually do it themselves these days. And yeah, so your natural bird is probably not that natural, but it probably doesn't have growth hormones anymore because that's not allowed.
I don't know. Do both.
Yeah. Anyway, we have, I think, thankfully made it to the end of our news for this week.
We have.
Yeah.
We lost Blair. She had to cut out. She had a hard out timeline.
I didn't know that. I didn't see that she did. I missed it. I missed the words. Yeah. Oh, she's sending mom home. Yes. Okay. Blair says, gotta go. Please send everyone my best. I'm sorry I couldn't stay. Well, we did go long today, a 2-hour show, 2 hours, 5 minutes at this point in time, I believe. Thank you, Blair. It was so good to see you. I hope that you have a wonderful Thanksgiving feast and maybe sit on the floor and eat with your hands.
Why not?
That's what I hope everyone
That's how I
does.
like to do it anyway.
Yeah. All right, everybody. Thank you for listening. I hope that you really enjoyed the show. I do want to give shout outs to people involved in making sure this show happens on a regular basis. Thank you to Fada for all of your help with show notes and with social media. That wouldn't happen without you. It's amazing. Thank you for doing that. Identity 4, thank you for recording the show. And Rachel, thank you for editing the show. Also, thank you everybody who watched live today, everybody who's in the chat room hanging out with us at this TWISgiving episode, which is off schedule and a little bit different than usual. Really appreciate you being here and chatting and taking part in the show. And as usual, I also must thank our Patreon sponsors for helping us keep this going. Thank you to Eden Mundell, Alan Viola, Aaron Anathema, Arthur Kepler, Craig Potts, Mary Gertz, Teresa Smith, Richard Badge, Bob Coles, Kent Northcote, George Corris, Pierre Velazarb, John Ratnaswamy, Quist Rosiak, Begard Chefstad, Donathan Stiles, aka Don Stylo, Ali Coffin, Reagan Shubrew, Sarah Forfar, Don Mundis, PIG, Stephen Albron, Daryl Myshak, Andrew Swanson, Fredis 104, Sky Luke, Paul Ronovich, Kevin Reardon, Noodles, Jack, Brian Carrington, David E. Youngblood, Sean Clarence Lamb, John McKee, Greg Riley, Mark Hessenflow, Steve Leisman, aka Zima, Ken Hayes, Howard Tan, Christopher Rapp and Richard, Brendan Minish, Johnny Gridley, Remy Day, G. Burton Lattimore, Flying Out, Christopher Dreyer, Artyom, Greg Briggs, John Atwood, Rudy Garcia, Dave Wilkinson, Rodney Lewis, Paul, Philip Shane, Kurt Larson, Craig Landon, Sue Doster, Jason Olds, Dave Neighbor, Eric Knapp, Lon Makes, EO, Adam Mishkan, Kevin Parachan, Aaron Luthen, Bob Calder, Marjorie, Paul Disney, David Simmerly, Patrick Pecoraro, and Tony Steele. Thank you all for your support on Patreon. And, we really can't do it with our Patreons, without our Patreon sponsors. It's all of you out there who allow us to keep doing the show week in and week out. So if you want to help support this show, please head over to twis.org and click on that Patreon link. On next week's show,
we will be back broadcasting Wednesday, 8 p.m. Pacific time, and then a second show, 5 a.m. Thursday, Central European time.
Same time.
Oh, yeah. I was wondering why I never got invited to the other one. We will be broadcasting live from our Twitch, YouTube, and Facebook channels.
And if you would like to listen to us as a podcast, just make sure to search for This Week in Science wherever podcasts are found. And if you enjoyed the show, get your friends and people at work, all sorts of people, friends and family over the holidays to subscribe as well.
Want to listen to us as a... Oh, wait, we already did that. For more information on anything you heard here today, show notes and links to stories will be available on our website, www.twis.org. You can also sign up for a newsletter. We love your feedback. If there's a topic you would like us to cover or address, a suggestion for an interview, a haiku that comes to you in the night, please let us know on one of our social media accounts or send us an email if you can find it. Just be sure to put TWIS, TWIS, somewhere in the subject line. Otherwise, your email will be spam filtered into an 80s sitcom starring Tom Hanks that's going to give you some very unscientific information about the dangers of listening to science podcasts.
And be forgotten by later generations, obviously.
Yeah. We look forward to discussing the science with you again next week. And if you've learned anything from the show, remember,
it's all in your head, this TWISgiving.
This week in science. This week in science. This week in science. This week in science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth. And I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got one disclaimer and it shouldn't be new.
there it was. The Obstacle Show.
Was.
Thank you, all live audience members, and then later unlive audience.
All the people who watched.
We want to thank all the dead people.
I will be setting this show up for rebroadcast at our normal showtime tonight. So if anybody missed the show, they'll be able to watch it later. But we've done it now.
I'll probably be asleep.
You better be. I hope that you get a good night's sleep. What time is it there right now? 10:30? 11:30?
10:30. Yeah, it's totally normal time here. I love doing this show at this hour. This is fantastic.
Well, we'll see. Yeah. Maybe Blair can do like a Wednesday noontime show or something. Some other time. Yeah.
Pretty awesome.
Yeah.
Because I have to wake up. Like, I wake up early anyway, but then I have to wake up even earlier and immediately be caffeinated and ready to do a show and talking intelligently at 5 a.m, which is,
it's great. I've been very impressed. I would not be able to do it. I don't know. Fada likes this 11 a.m. time. I know. I like the 11 a.m. time. I know we had a couple of people who said they were watching in Europe for the first time, like in England, they're watching because this was a great time for them to watch.
Yeah, because who wants to get up that early?
I know. I think it's our. I don't know, everybody. Let's talk about a daytime show. Can we talk about it at some point in the future? Let's see what we can do. I think Blair likes to have. Evenings are hard for Blair too with the child.
All the time, all the time is difficult when you have a very young child.
That's absolutely true. Mm-hmm.
Yeah, this is the easy time.
No. Even the sleeping times, you're like, something's gonna happen. Oh, but you're late, and it's we've been on the air for a bit over 20, not 20. Geez, what is my brain
20-hour
doing?
show.
More than
Another
20
20-hour
minutes.
show. How do we do it? I don't know.
Turks,
Persistence.
9:30 in Ireland. It's great. Yeah.
We'll get to the science news through attrition. That's how we'll do it. Okay, well, in that case, maybe it's time to say, say good day, Kiki.
Good day, Kiki. Say good night, Justin.
Good night, Justin.
And good night,
minions.
everyone.
Good night, everybody out there.
Good
Good night,
day,
patrons.
everyone. Thank you so much. Have a wonderful week. If you are in America, United States of America, have a wonderful Thanksgiving holiday. No matter where you are, spend some time giving thanks this week, because it's always good to have a little gratitude for the science in our lives.
I was unable to find a turkey.
No.
I don't
Have a pork. Have a pork loin. I don't know.
Yeah,
Have
wings.
a game hen. Have a salad.
So,
Go pick some berries and sit on the floor and eat them with your
pork
child.
meatballs is probably the most.
Okay, everybody, have a great week. Stay safe, stay healthy, stay curious, and stay lucky.
Gotta stay lucky.
Yeah, we'll see you next week. Normal time.